I really get bad. Yeah. All right. Well, the fact that plants contain compounds, not just calories, not just macronutrients that help, strong bones and teeth, but compounds that actually will interact with human therapeutic targets to counteract chronic disease or to improve metabolism. That's been known for quite some time. That's been known almost since the beginning of recorded history. The most famous quote is probably 460 BC, Hippocrates. Let food be thy medicine and medicine be thy food. So that's been, that was maybe the most famous quote, but it even goes back further in the evidence than that people were using foods, not just to eat for calories, but to eat for medicine. And there was almost no distinction between the two. But it's just within the last couple of years that people are getting more and more realizing that foods, that they need foods for health, and they need foods for immunoprotective plant bioactives. And people are getting more and more in tune with the fact that plant bioactives can improve their immune health. Now, most of us, we've lost that. In the modern times, we've kind of lost the connection between what is the food, what is really in the food that we eat. I mean, yes, you can order a steak in the restaurant and have a side of grits or a side of rice, and you know what plants and animals those are from. But a lot of the processed food, we've lost it. Like, what's really in a Twinkie anyway? Do you know? Not real good. I mean, they're practically indestructible. So there's a lot of preservatives in there. There's animal tallow in there. It was only about 8 months ago that my son was feeling guilty because he had a Rice Krispie treat. And I'm like, and he's a vegan. And he's, oh, it's terrible that I did that. And I'm like, what is not vegan about a Rice Krispie treat? Well, the marshmallows have animal tallow in them. It didn't even occur to me. So we just kind of lost touch with what is in our food. But it's just in the last couple of years that we got more and more, oh, what did I do? All right. You showed me how to use the pointer. Yeah, there we go. The immune protective properties of plants. People got really interested in that in the last couple of years. Why? It's been this 400-pound gorilla in the room. It's been COVID, right? Everybody knows you get COVID and you can survive it unless you're immunocompromised. And all of a sudden the focus came on, okay, we can improve our immune protection by drinking elderberry syrup. We can improve it by certain foods that help improve immune health. And that's kind of put the focus back again on foods, certain foods that help to protect the immune system. But that's why we care now about nutrition and the effects on immunity. It's a crisis that has really shaken up the way we think about food, the way we think about a lot of things, like consumer trust or not in science. Is Dr. Fauci a well-respected scientist, or is he a minion of the devil? It goes both ways, right? You hear both things. Consumer priorities for nutrition advice and products, consumer views on nutrition in the context of globalization. We're used to getting our food from all over the place now. Like, we used to only get strawberries during the season. Now we get them from all over the world, and we can have strawberries all year long in the grocery store. But is that a good thing now, we think, with the borders and the immune, the regulations are different from one country to the next. So people are starting to rethink things. So the way we eat, what we eat, nutrition, lifestyle, how we prioritize nutrition and nutrition products, it's changed. It's changed because of COVID more than anything else. It was changing even before COVID, but that's really brought it to a head. So we know now if we're immunocompromised, we're at more severe risk for COVID, but probably whatever's going to come after COVID, right? Because it's going to keep happening with a globalized society. So we're more worried now. We're more in tune with what we eat. A good friend of mine, probably my best collaborator, David Neiman from Appalachian State University, he wrote an article in 2020 about the coronavirus as a toxin to our aging, corpulent, unfit society. Toxin, not Toxin like a poison, but toxin like a bellwether, like a wake-up call that, hey, we're doing this to ourselves. We're making ourselves vulnerable to any disease that's out there. Immunosenescence is the natural loss of immune protection as you get older. It just happens. And one of the best ways to alleviate, to mitigate immunosenescence is the combination of the dietary intake of foods that help to protect the immune system and moderate physical activity. We'll talk more about physical activity in a little bit. So again, the two things that we want to do is we want to eat right, we want to eat healthy foods, and we want to have good physical exercise. These are two things that we have complete control over as individuals. We're not depending on anyone else. We can do it. We have the ability to make ourselves resistant, except medicine is no good unless you eat it. And everybody knows that. Everybody knows eat your fruit and vegetables. Everybody knows immunosuppression, but you can't just tell people that. You have to deliver. You have to make the foods that are immunoprotective, that are health protective, you have to make them palatable. You have to make them easy for people to eat. You have to find ways to do that. So we have the FDA regulations, US dietary recommendations. And you all know you're supposed to eat 5 cups of vegetables a day, fruits and vegetables, and 13% of the US population does it, if that. So they're just not doing it. So what we do eat is like 1.8 cups a day, and what we're supposed to eat is the 5 cups a day. It's very, very seldom. What are the excuses that people use for not complying with what they know is going to be best for their own health? It's inconvenient. If you buy fruits and vegetables at the farmer's market and bring them home and put them in the bottom of your refrigerator crisper, you'll forget about them. They'll get wilty. You have to cook them, big pain in the neck, much easier to buy a granola bar, right? Perishable, that's another thing. Expensive, people consider fruits and vegetables more expensive, and sometimes they are, than McDonald's. Taste is a big one, and bulk. So these are reasons that people do not want fruits and vegetables. So we've got to change the way we do things. Now, where am I coming from when I'm telling you this? I want to explain where I'm from, and Andy did a good job of that in the introduction, but I'm from the North Carolina Research Campus, which is a campus with 8 different universities on it, all dedicated to human health, natural products, plants, biotechnology. So, most of your universities, like here, you'll have history, you'll have law, you'll have engineering. No, we don't have that. We just have, everybody's focused on human health with these 8 different universities. And you see that dome that's on the top of the core laboratory, big copper dome. This is a 311,000 square foot core laboratory that we have on campus. And that dome on the top, if you look into the dome and you look at it from the ground level, it's all fruits and vegetables. It's a big mural of fruits and vegetables and an eagle. I won't get into why that eagle's there. Ask me later, but all fruits and vegetables. You don't really get a feeling for, so we're serious about fruits and vegetables. We're serious about plant foods and their impact on human health. You don't realize how magnificent, this is, going 4 floors up, you don't realize how huge and magnificent these painted fruits and vegetables are until you see the artist next to them. They're just magnificently huge fruits and vegetables. And that's really the theme of the whole campus. We are located in North Carolina, kind of inland, like halfway between the mountains and the coast in North Carolina, very close to the Research Triangle, the Chapel Hill, Duke, NC State main campus, and Wake Forest up on the northern part of the state, and then some of the medical facilities and science campuses in South Carolina. So we're kind of in the middle of all that, kind of reaching out, our place in the world, reaching out to try and be a center for the importance of plants and human health and natural products and, some of these other alternatives to the pharma industry that, or even adjuncts to the pharma industry that can improve human health. So again, we are a public-private campus. So we have the 8 universities, we have a community college on site, we have different industries that are partners with the campus, many that are physically on site with the campus, everything from the big players like Standard Process and Dole to small entrepreneurial CBD makers, things like that are on campus. So we've got a whole range of different industries like that are on campus that are partnering together. And we are famous for the omics. So, we do the genomics, the transcriptomics, the metabolomics, the nutrigenomics, bioinformatics. We do the gamut. And whether you're looking at an animal system or you're looking at a plant, the same omics techniques can be used to generate data to find out what you need to know data-wise from these different systems. Okay. And the work that we do, we try and be transdisciplinary. So interdisciplinary is when you have a project and you set it all up and you kind of plan with, let's do a project on this. And then somebody says, oh, we really should have an economist. So let's ask an economist in. Transdisciplinary is more like you do it from the ground up. Like before you think of what your hypothesis is going to be and what your project is going to be, you get all the brains together. And some of the ideas might be crazy and seem stupid, but you put them all together before you even start putting the hypothesis together, and you build your research programs that way. So we'll have the horticulture in there and the bioactives and nutraceuticals and the breeding, the functional genomics, all the way through food science to human nutrition, which, and different universities are specialists in different areas. Personalized nutrition is actually UNC Chapel Hill, but we interact with them quite a bit because people at UNC Chapel Hill are fantastic with nutrition and understanding the human system and responders and non-responders, but they may not know a plant from their foot. They may not, they may think, oh, I can buy this compound off the shelf and it's from a plant, but they may not understand why a plant makes that compound, what the relevance is to human health. And so it's a really good partnership to put it together. We are this little building in the beginning, in the front. We're the Plants for Human Health Institute. I actually took this picture they had a helicopter flying around the campus. It was some kind of fair, and I got to take pictures. And I always tell people I didn't cut off the Chapel Hill building on purpose, even though we're terrible basketball rivals. But this is us, and it's 110,000 square feet, and we're mostly North Carolina State University, but we also house Appalachian State University, the Exercise Performance Laboratory. We house a little bit of UNC Charlotte in there, and North Carolina Central University. So we're partners in a lot of things, and we share facilities, and all work as happy campers as much as we can. And our motto, or our mission statement, I guess you'd say, is leading the discovery and delivery of innovative plant-based solutions for human health. So not new news, but figuring out why this works. Not just, apple a day keeps the doctor away, but why? What the heck is it? What interactions between compounds and plants are contributing to a change in an enzymatic system in the human body, or metabolomic flow in a human body? We work everywhere from whole foods and functional foods all the way to topical formulations, but mostly we're most on the functional foods, whole foods end of things rather than phytopharmaceuticals or ingredients. We do it all, but mostly on the whole plant level. Okay, and then part of the campus, and back to that core laboratory that I showed you before, this is brand new. So part of the core laboratory now is the North Carolina Food Innovation Lab, which just opened in 2019, October 2019. So, COVID, but still it opened. And what this is a facility, an innovation lab, totally dedicated to to processing plant foods to preserve the bioactivity. Now, most processing, no, they're interested in preserving the color, preserving the flavor, preserving the shelf life, keeping out microbes. This is doing all that too, but preserving the bioactivity that you expect from the plant that's the food source. And it is a facility that, the vision, it was funded by the North Carolina legislature with the idea of adding economic value to North Carolina, because North Carolina, we make a lot of crops, a huge variety of crops. We're the #1 sweet potato producer in the world. Muscadine grapes, all other crops like that, small crops, but a lot of these crops are grown as fresh market or shipped out for processing elsewhere. And so this was an idea of, all right, what can we do in North Carolina to keep the processing here and to add value to these crops? And then the mission is to have a collaborative environment where we can work together with academic scientists and industry to accelerate plant food-based research and development. It's not, so unlike a lot of pilot plants that are in universities, this is not a teaching lab. So we don't have like classes come in and learn how to use microwave processing. We don't have that. It's, for one thing, our campus is 100% research. So we only have graduate students. We don't have undergraduates at all. We have undergraduates swarming the campus during summer and sometimes breaks when they do internships at our lab, but mostly it's graduate students, technicians, research associates, postdocs at our campus. And it was put together because the food industry really is changing. We're more into health and wellness. We're more into green systems, being able to read a label and know what's on there. People are more concerned about that than they ever were before. And there's been a huge jump in food sales just in the last couple of years. Plant-based milks that I guess you're not really allowed to call them milks anymore, but plant-based milks and plant-based meats. And this was really provoked by COVID. It helped to provoke the growth of all these plant-based solutions. It wasn't there before, and now it's huge. Now that the sales of some of these things is just skyrocketing. So the processing lab will go everywhere from the raw commodity to traditional products, like, somebody in North Carolina has a great barbecue sauce that they think is better than Texas barbecue sauce, and they want to market it. They'll never make it, right? But they'll want to do that, so they'll come to the lab. And then the big processors too, the General Mills will come, Progresso will come, because they've got a new secret formulation. They want to do a small run to see if it'll work before they convert their whole facility. And a lot of novel ingredients in processed foods. And a lot of the processing equipment in there is all geared toward health benefits of the food. Like one of this is this continuous multi-frequency microwave, which is something that I work quite a bit with in some of my technology. And this is a way to process foods without a lot of heat. Well, it's heat, it's microwave heat, but it's continuous multi-frequency. So you're pulsing heat to preserve, to process and to either pasteurize or actually preserve a food, but without reducing the flavor, the aroma, the color of the food. I think I have to go over here for this. So for example, this is actually one of my technologies that they just put on production, where they're making like a baby food. And one of my technologies, which I'll talk about in a minute, is taking protein, combining it with polyphenols to make a colloidal aggregate particle. It's both of these. It stabilizes the polyphenols, and they're using it in a formulation for baby food to get some of these active compounds in a very stable way into the human system. Done it in a lot of other things as well. So again, the Food Innovation Lab, it's not just a pilot plant. It's got an ideation center, pilot plant production, product development, all the way to sensory. So we're trying to do all that in one place. And a lot of work is actually that middle one. Food photography is something that a lot of people leave out, but making the product look good, making sure you emphasize what it is about the product. That's an important part of it if a product's going to make it. It might taste great, but you've got to make it look like it's going to taste great before somebody will buy it. So they'll have all those things in the Food Innovation Lab. So all these things are geared toward preserving in a processed food, and hopefully in a smaller volume, all the benefits of the food so that you can eat it, maybe in an ingredient so that you can eat it. So you get all the benefits of eating a spinach and arugula salad for lunch every day without having to eat the spinach and arugula salad for lunch every day. That's kind of the point, is how do you make it fun, tasty, smaller volume than having to eat a truckload of spinach? I mean, Popeye's great, but that's too much spinach. So you just got to find some way to get it in a smaller volume and tasty and good. So what we're all about is we're trying to decipher these phytoactive components, sort out the interactions between them, because no plant works with a single magic bullet. It's always interacting compounds. And then decide how to deliver it in a health protective fashion. So if we look at the difference between foods and pharma, if you're sick, you have a condition, the pharma approach is to go to the shelf, get a medicine that's going to take care of that illness, right? So the way that pharma works is you have a single active ingredient, goes through high throughput screening so they can test it to make sure that a certain enzyme system is disabled before you eat the thing or before you take your dose. And it's always a defined dose and a defined clearance through the human body. And plants don't work like that at all. They have multiple phytoactive components, interactions, synergies, hopefully, or at least interactive effects between the compounds. And sometimes we don't know the metabolites. We're trying to get to them. We're trying to figure out what they are. Doing better with metabolomics now, human metabolomics, to figure out when you eat a tomato, when you eat a berry, what are the metabolites that surge through your bloodstream, and how can you modulate that surge of metabolites through the bloodstream? How can you control clearance through the urine? How can you do that in the human body to get the more bang for your buck? Now, a lot of times a plant extract, when you have a plant extract, maybe let's just say something that reduces blood glucose, and you have that plant extract, and you will take that complex extract and you'll separate out different components, separate until finally you get the big bang, and you know that material is the material, that is the compound that does the job, okay? And that's what we got. Oh, did it again. Okay, that's the compound that does the job. So you've got an isolated compound or maybe just a little trio of compounds, and that is the anti-diabetic compound. But what we very often find is you get to the bottom and nothing happens. And so what you have to do is have a synergy. You have to have more than one compound together, usually a lot more than two, to get the biggest bang, to get the biggest health protective effect, because that's how plants work. That's how they work for their own protection under stresses of the environment. And that's how they work in the human body as well. So I'm talking mostly about plants that are, well, one of the original sources, the original source of plant bioactives is wild plants. That's how we started. That's back when Hippocrates made his statement. He was talking about wild plants or lightly farmed plants. And things have changed. We've gone from, and I'm not saying cropping plants or farming is a bad thing, but it has changed, okay? We went from wild foods to modern cropping, and that gave us higher yields when we do higher cropping. It fed the world. We are now able to do shipping qualities that allow us worldwide distribution. Consumers want things sweet, so things have changed with breeding. So we're tending to have our fruits and vegetables be sweeter and or more bland, because sharp flavors are not, especially with the American palate. Americans are terrible. They just want, stuff that doesn't taste like anything and put a lot of salt on it. Like Doritos with all that orange stuff that they put on it. I mean, the Dorito by itself would taste like nothing, right? But they put, that's just our American palate. And so we've changed how we process the food, how we make the food, how we breed the plants. Consumers also demand perfection. So we have a lot of waste material that comes out of things, and farmers want efficiency. So we've done all these things to our wild plants to convert them to modern farming. But the negative outcomes is we rely a lot more on inputs. So we have to do fertilization, pesticide application, plowing, all those inputs to the field. Again, not a bad thing, but it has changed how plants are processed, how plants are grown. And it has depleted some of the nutrients, because every time we breed in a little more sweetness, we're breeding out an active compound that might be bitter, but also might be health protective. And we've diluted those phytoactive compounds. So when you go to the grocery store, and I'm not saying this is bad either, this is great. More people should shop in this part of the grocery store, not the other parts with the ice cream and everything else. They should shop in this part. But most of the plants that come from this are from high production fields, like this strawberry field in California. Great, but it's very, very different than this field of wild berries in the tundra in Alaska. And this is one of the places that I work in. This is Point Hope, Alaska, which is a scary place. I'll tell you about that later too. So it's a wild field, and berries is all that grows there. This is a coral berry that you see there in the lower corner. And, but it's different. So they're abundant, they're prolific, but they tend to grow like a lot of things together. It's not going to be a monoculture of one berry. It's going to be whatever's deciding to grow there. So we have blueberry, tundra cranberry, and coral berry all together in that upper photo. And a lot of times the wild plant in the tundra, it's a tremendous, tremendous berry because it's under 23 hours of photo period. This is Alaska. So it's under the extremes of cold. I was just there last month, 14 below, 9 feet of snow. These berries have to put up with that in the winter, and then in the summer, 23 hours of photo period during their ripening season. So what happens is they may not make it to the ripening season. Here you got this berry going into fall color. This is a Vaccinium uliginosum, which is the bog blueberry. It's very famous in the tundra. Got a lot of medicinal properties with Alaska natives. And it's going into fall color. It's never going to make it. It's not going to ever be ripe before the first frost. Well, it's already had the first frost, but it's not going to make it before the snows cover it. So sometimes it doesn't make it. I'm not saying that wild foods is the answer, because we couldn't live. We couldn't have our population with just wild foods, but things have changed. So wild foods tend to have, without the inputs, they thrive without the inputs, and they're underappreciated for their health properties. A lot of these compounds, flavonoids, stilbenes, phenolic acids, lignans, they're more abundant, richer in the wild plants. We need to find ways to keep these in the cultivated plants or breed them back in. And a lot of effort is going on now to try and do that, to try with genomics to breed these things back into some of our popular crops. So this is a wild blueberry field, a piece of tundra with several kinds of blueberries, blueberry, cranberry, coral berry in there, and Kelly Clarkson. So what do they have in common? Kelly Clarkson, okay, first American Idol. She was on The Voice for a while. Kelly Clarkson show now. What do they have in common? What was her signature song? What doesn't kill you makes you stronger. Live. Okay, so that's how the berries are. They're the fringe of survival. They're going through a lot of stress. That's what makes them stronger. That's what makes them much more potent for human health. And we have changed a lot of our crops through breeding. Again, it's something we had to do just to survive, to get the populations we have now. But this arsenal of interacting phytoactive compounds is a plant's passive aggressive strategy to withstand the environment. Plant can't get up and run away, right? It's got to put up with every insect, every frost, every UV light. It's got to put up with it, and it does it in it. That's the only thing that it can do. So it's got to make chemicals inside of it. We've done. There's not a lot of direct comparisons, by the way, between a wild plant and a cultivated plant. So you can directly say, oh, the wild has more of this, and because they've changed. Sometimes it's just not the same species that is now cultivated in the field that's in the wild. And most of the times it's not the same species, not the same genotype. You can't make direct comparisons, but of those that have been made, you'll find differences in mineral content, differences in phytoactive content. And color is actually an easy biomarker for bioactivity, because it's one of, the color, the anthocyanins, the carotenoids, the glucosinolates, the betalains, those are kind of biomarkers of bioactive compounds that actually improve human health. And those same pigments that the plant naturally accumulates to ward off the environment, those are the ones that interact with human therapeutic targets to ward off disease. So take an example, the blueberry. And we actually talked about this with a couple of the meetings that I had. Cultivated blueberry, if you cut them open, and hey, cultivated blueberries are great, eat them. You'll live a long time if you eat them, but you cut up a cultivated blueberry, it's be yellow or white inside. It just will be. Cultivated blueberries are only about 100 years old. They were developed in New Jersey about 100 years ago. And now they're actually worldwide distribution and growing in other countries. But a wild blueberry, it's like this, okay? You cut it, you pick them, your hands are gonna get red. It's just how it is. They're chock-a-block full of these phytoactive compounds, and it's sort of like a beacon, that it's got the bioactivity that's maybe not there in the cultivated variety or the cultivated form of the blueberry. And the term, you all know this because you're in horticulture department. So the term is elicitation. So when you stress a plant, it elicits the production of phytoactive compounds in the plant. Environmental stress will elicit this. This is a wild blueberry field. Wild blueberries grow wherever the heck they want to grow. You cannot tell a wild blueberry where to grow. You cannot transplant a wild blueberry someplace else. It grows where it wants to grow. If you own a field that has wild blueberries, you're doing very well, but you cannot buy a new field and decide to grow wild blueberries. It is a species that, like, if there's a fire and the forest burns down, the wild blueberries will start to creep in and colonize and take over after the ashes are gone. But it's a natural thing. And elicitation is part of what makes these blueberries so potent. So when you pick them, your hands are going to be all red. When you exercise and you can't just take something little like this and pump it and say, I'm gonna get big muscles from this, right? What's the expression? Okay, no pain, no gain, right? You have to hurt yourself. You have to have a big weight and do it repeatedly and have a little pain before you have the gain of big muscles. So the similar thing for potency in a wild plant, no stain, no gain. It's just how it is. That is one of the biomarkers of where the active ingredients are. And by the way, this is it's not just true of the plants, not just true of the horticultural plants or the wild plants. It's true of the production of meat products as well. Like the chicken used to take 16 weeks to go from an egg to a 2.2 pound fryer chicken. Julia Child would write about how to fry this chicken up with a little butter and a little salt, and it would taste great. The same chicken now the chicken takes 35 days from an egg to 3.7 pound big chicken with breasts so big that it can't even stand up sometimes and totally misshapen. And the same chicken doesn't taste as good. Julia Child was, you guys know, you're probably too young to know Julia Child, famous French chef, but she was appalled that how chicken that she had written about in her French cooking, the joy of French cooking cookbooks, she could no longer make those recipes because the chickens had changed so much because of breeding. Again, it had to happen, probably had to happen to support the population, but it has changed the chicken. They don't have the lipophilic vitamins and content and flavor that they used to have. So we know we need nutrients. We know we need phytoactive compounds. We know we also need exercise and movement, because what the research has shown, and this is just in the last few years, is that movement, the physical active movement, especially vigorous exercise, but even mild movement, will help the bioactive compounds that you eat get down to your microbiome, because a lot of them have to be metabolized or catabolized by the gut microbiome into smaller metabolites before they surge into your bloodstream and circulate and get to the targets where they need to. So movement is important as well. We just are in the final year of a project with the Foundation for Food and Agriculture Research, which is a lot of fun. It's a great agency because they fund things that are broad and out of the box, where we're combining, see if I don't turn it off again. We're combining some of the breeding genomic techniques to look at what makes different plants that have compounds more bioavailable in the human body. And we're looking for product options and ingredient options that will take those bioactives and deliver them in a food product so that we're pretty sure they'll get into the bloodstream, that they won't just be excreted in the urine and do no good for your body. Because a lot of things do. Like, you can, people will like buy these 1,000 milligram vitamin C tablets and they think they're doing good for their health, and all they're doing is making expensive urine. That's what people are doing. And you really have to know if it's going to get into bloodstream or not, if it's going to do good. And then defining nutritional equivalencies. So we're trying to take these foods that we deliver in different ways and make sure that the amount of an ingredient will give us the nutritional equivalency of a serving of the fruit. So like one of the things is we've made a protein polyphenol particle, taking phytoactive compounds from berries or spinach or kale and combining it with protein. I'll show you that in a minute, and making like a flour, and then putting as much in 25 grams of that flour as you'd get in a whole 175 grams of blueberries. So you can eat it, lower volume, better flavor, or good flavor in a product. So that's part of what we're doing, because it really, honestly, it doesn't matter how much is on your plate, like how much anthocyanins are in an apple. It really matters if those anthocyanins in that apple will get into your bloodstream. And different varieties of the same crop will differ in their ability to be metabolized, to be absorbed. They just have different abilities. We worked in this project with 3 different crops: blueberry, banana, and spinach. Blueberry, because I do blueberries. I'm the blueberry queen. And then banana, because Joel is one of our partners on campus, so that was a no-brainer. And spinach is one of the leafy greens that is quite popular in the US diet. And we had diverse germplasm collections in each of these. We used UC Davis for the spinach collection. Banana, we had the whole Costa Rican population of dole, looking at different genotypes, how they differ in if we look at the genetic composition and then compare that to how bioavailable they are, doing in vitro bioavailability screens or in vivo in animal studies, we'd find quite a few differences between different genotypes, whether a spinach had a wrinkled leaf or a smooth leaf, whether, the different leaf shapes, which probably wasn't the important thing, but different varieties of spinach would just be different in how well they were absorbed, how well those carotenoids were absorbed into the system, how well other chlorophylls were absorbed into the system. So that's part of what we're at. One of the things, and I just told you about this a little bit, is that we take active compounds from berries or other, we've done rosemary. The lab smells great when we do rosemary. But we take the active compounds and we fuse it with protein. It could be usually a plant protein. We've done soy and rice and chickpea and protein isolates, very high protein isolates. This happens to be whey protein. And then we fuse them together, complex them together in a process, and either freeze dry them or more commonly spray dry them to make it's like cake flour, except it's colored. And what it does is it absolutely preserves the bioactivity that's in the original fruit. But if you ate the original fruit, you get it in your mouth, it goes down the esophagus, it goes down into the stomach, pH differences, enzymatic differences, a lot of it's going to be degraded before it gets down to the colon. This protein complexation protects the phytoactives so that it gets all the way down to the colon and has a chance to be catabolized into the active metabolites that can then surge up into the bloodstream, especially if you exercise. So we know that, the dietary polyphenols, they got to get down to the colon before they can be made into, converted by the colon bacteria into simple phenolic compounds, which then get up again through the translocated via the portal vein in the liver, and then get out again into circulation, phase 2 conjugation, into smaller metabolites that are actually, we have shown in some of our studies that the smaller molecular weight metabolites, catabolites of the big anthocyanins, for example, are more bioactive in vitro studies and in vivo studies than the anthocyanins themselves. It's the metabolites that actually do the job. And we combine that with exercise in a lot of our work, because they really get released into circulation and surge into circulation when you're moving. If you're a couch potato, not so much. Okay, so and this is an example of rosemary complex with plant protein rather, and well, plant and animal protein because there's some whey and some chickpea there as well. And how the compounds will get down to the colon only with exercise. So that these aggregate particles don't degrade in the gastrointestinal tract, which is a great thing. What we like to do is use pumice. It's a waste product in the industry mostly, although some people are now starting to grind up pumice and use it in, actually sell supplements with pumice because the health value is being recognized. But it's the seeds and the skins and the leftovers after the juicing process, and we'll extract from that pumice using that microwave processing that I was showing you in the Food Innovation Lab, and combine it with protein so it makes a colloidal aggregate. It can be either a thick slurry like this, or we can have a dried particle like a flour. And they're great for clinical and preclinical applications, because if you're going to feed somebody, you're going to get a group of people together and say, we're going to test the effect of spinach on muscle damage. They don't want to hear that they have to eat this much spinach, right? They want something that's reasonable that they're going to have to eat every day. So it's been great as a preclinical and clinical intervention. Preclinical, we've used a couple of different models. Now, one that we love is sled dogs. The sled dogs will eat anything. They're great. They'll just put it down, it's gone. But if we do it in this protein polyphenol particle, it gets into their gastrointestinal tract and it makes it all the way down to their colon. And we combine that with exercise. So we use these dogs. Excuse, this is going to be a little noisy. But these dogs are crazy. And they love to run. Short intensity endurance dogs. And they are great for us because we'll feed them. We'll take them on a sled ride, and then we'll take blood from their forearm and be able to measure that blood and what the effect of blueberries or salmon or whatever it is that we're feeding the dog, but in this case, it was blueberries, which was just last month in Alaska. Measure the effect on inflammatory markers, the ability of the intervention to depress the inflammasome activation in the dogs, prevent damage in the dogs. These dogs are very easily damaged. They're not Iditarod dogs that run the big races in 8 days in the cold or 2 weeks in the cold. These are dogs, short duration dogs that actually run short races, but they have very exposed testicles, and their ears are floppy, and they can't make it in the cold for a long period of time. They're just short duration dogs, but they're actually bred for that. And so they want to find every way that they can to prevent damage on these dogs. Another model we use is neonatal pigs. Actually can get to be a handful. And the neonatal pigs, doesn't want to go. There. We use them for allergy because, I said we take phytoactive compounds and we combine them with plant protein. Well, these plant proteins are very often allergens, right? They have allergenic epitopes on soy, on whey protein, and peanut protein is a big one that we do. And when we complex it with the polyphenols from plants, from berries especially, cranberries especially, it changes the conformation of the epitope such that when you eat the peanut, you don't get the IgE turned on. You don't get the anaphylactic shock or the allergenic reaction. Your T cells do turn on though when you eat it. So you're getting the protection against the allergy without the allergenic reaction, especially good for oral immunotherapy. You have a kid and he eats something and absorbs the protein, and IgE will bind on the mast cells, but then the next time he eats the protein, he's going to get this hypoallergenic, this allergenic response. And so our epiphany was, could we combine cranberry with peanut protein and make something that's hypoallergenic? And yes, in our tests we have. And with the pigs are so great because they get allergies just like people do. Mice really don't. Rats, we've done rats. We could get some results from rats, but they don't get allergies like pigs. And pigs are great because they get the same symptoms. They get vomiting, they get diarrhea, they get the skin reactions, they get infiltration of eosinophils in their esophagus. So these little protrusions in their esophagus in reaction to allergies. So you can get those same allergenic reactions and you can measure the ability of the intervention to cut off that allergenic reaction. So a couple of animal models, but here we're showing, in this case, peanut flour with cranberry in a 1-to-1 dilution to make those particles actually depresses the allergenic proteins quite a bit. And digestive fragments of those peanut proteins also, same thing, they're less immunogenic. But the best model, of course, is humans, and we've used all kinds of, and we will use overweight, obese, postmenopausal women in some of our trials, but the best is to use these high performance athletes, or at least weekend warriors, because they're really great. They're so motivated and compliant. They want to do the trial. They don't care if you take a biopsy out of their leg. They say, if it's going to make me run faster, go for it. I'll eat whatever you tell me to eat. They don't like the postmenopausal women. I'm talking about the down here. Okay. They'll be like, oh, I think these blueberries are making me have diarrhea, or I forgot, or, I'm going to church and I'm going to the potluck and I don't want to ruin my appetite. And, they'll just go off the program. But the athletes, no. Athletes are going to do it if it can possibly make them better. And they're great. And athletes are actually highly susceptible to infection. And we work a lot with DOD and warfighters as well. So we did this one, much of my time, we did this one clinical where we supplemented athletes for 17 days with a protein powder. It was soy protein in this case, complex with blueberry and green tea. So we had a high, it was equivalent of 2.5 cups of blueberries a day and 2 cups of brewed green tea. So not unreasonable, but in 25 grams, okay? So the athletes took this every day for 17 days, 14 days of supplementation, and then they began exercising. 3 days, intensive exercising, 2.5 hours. Polyphenolic signatures were post-colonic, were in the serum, were incredibly enhanced when they ran. Like they were taking blueberries or taking this supplement with polyphenols for 14 days. Yeah, a little bit would get in the bloodstream because we're taking the blood draws. But once they started exercising, boom, it just starts surging. So that, because we didn't let them exercise or just moderate exercise up until then. And then enhanced ketogenesis. So they were burning fat for days after the exercise. It just kept going when they took the polyphenol supplement, which is a good message. So their signatures were enhanced. Like even after recovery, they were having a blueberry signature. Arabinose is a sugar that's on the blueberries. It was enhanced in recovery. It was enhanced in workout if they had that supplement. So it showed that the athletes were immunocompromised actually, but that the blueberries actually improved their performance. You can do it with walking too. You don't have to kill yourself 2.5 hours running, but just moderate walking will be enough to increase the levels. Like if you had runners and see how their flavonoids went up, but even walking, their flavonoids went up. If they sit there like a couch potato, even though they've had polyphenols, it's not a lot in the bloodstream. In fact, it's even negative in this particular one. And then the placebo walkers, just people taking placebo and walking, it just enhanced the natural flavonoids that you get from everything else you eat, because you get flavonoids in every plant food that you eat, almost. Even that was enhanced in the bloodstream. So walking made a huge difference. Exercise made a huge difference. So eat, move, think. We're proposing to the NIH a study where we can look at these different scenarios of having elderly people with blueberries and walking, and just 45 minutes a day, in a social gathering, and how that improves their cognition. because we know that when the berries get down to the colon, it shoots up those phytoactives, which do pass the blood-brain barrier and improve cognition in the elderly, improves memory, improves retention, recognition in the eye. Very microseconds difference, but enough between the ability to drive or not drive. And our control groups have stretching because that socialization in the intervention, the 45 minutes of socialization, that's part of cognitive enhancement. So we wanted to make sure that that's equal. So you can't just be a couch potato, even if you are eating good things, can't just eat, you have to move as well. One of the big things that with exercise, with athletes though, it's not really running faster that matters. It's that the athletes are tremendously immunocompromised when they get done with a marathon. It's just the way it is. So we took those same athletes, and each of these red lines, each of these red arrows is a blood draw. We took those same athletes and we took the blood that we drew at different times and did a serum antiviral assay with it. And we found that we took human cells, human HeLa cells, and added serum from the athletes to the cells and monitored that. And we found that when we fed the athletes the intervention, the polyphenolic intervention, the cells, the viability of the human cells stayed alive. So the virus was less active in killing human cells. And in the same way, the virus was less able to replicate, which is a problem when you get infected with virus. You want to cut down the replication. So the virus was less able to replicate in the intervention when the athletes had a polyphenol supplementation. So that was our conclusions, that the athletes are totally vulnerable post-exercise. It's well known now that, athletes are one of the worst as far as immunocompromised right after they exercise. People do the marathon and get sick that weekend from the flu or something that looks like a flu because they do get immunocompromised. But use of a polyphenol protein supplement improved their ability to reject viral infection. So all we need to do is get people to exercise more and eat functional foods. We all know that's not so easy, right? It just doesn't happen that easily. So what do? That's our job. We've got to make that natural association between polyphenols and proteins, make particles that are delicious. We've done some work on that. We've done some work on the sensory applications. We made a lot of different products out of it, modified to fit your application. We've done work on, I've showed you the microwave system to extract from pumice to get the value added from waste materials. We've made some delicious gummies, which mothers think are a sneaky way to get protein into their kids and polyphenols into their kids without sugar, because you don't need sugar. When you put these two things together, it modulates the flavor. So you really don't need sugar supplementation. It tastes good. It tastes like the fruit, and it modulates the protein. So you don't taste any beaniness like in a soy protein. You don't, because it's put together. So it's just a sneaky way to get the good things inside in a small volume. And that's key, small volume, because people don't want to hear that they have to eat 5 cups. Who can do that? Really? Think about 5 cups. I can't. So it's great. We've worked a lot with the military because this is important for the military, especially, having something portable that you can, put in your backpack and get out into the field. You can't bring fresh fruits and vegetables out in the field in Afghanistan. Well, not Afghanistan anymore because they're not there anymore, but wherever they are in the field, they can't do that. So we work with the military to make supplementation that they could bring in the meals ready to eat. But you don't have to be military. You might be just, you might just want to be a backpacker, that needs a easy to eat supplement that's shelf stable. You might be the guy in the backpack. Who knows? You could be anything, but you just, just to have that for normal people, have a supplement that gives you that nutritional and extra nutritional ability is key in a small volume of material. We've also worked with Bill and Melinda Gates in Zambia because, make sure, we want to make sure that people in Zambia during the dry season, they're just eating cornmeal, nishima. They don't have ability to store fruits and vegetables. They don't have electric refrigeration, so they can just mix this into their nishima and get those phytonutrients and protein, or just regular people who just need, they need that little edge. They need something that's not going to be mounds of food, but give them the nutrition that they need. So American Public. And that's all I have for you. That's our institute, and I'm happy to answer any questions. Sorry, I'm just right at the edge here. Thank you very much. Yes. Oh, I'm sorry. Yes. He's asking about with this particular or just in general, because it is rife in the industry. Oh my gosh, yes. In fact, you know, like we know we published something and it's our lab, and all of a sudden, a few months later, it's published in other countries. We know they didn't get that idea, you know, all of a sudden, and they're not doing it, but it gets published. So yes, it's a big problem. There's a lot of problem with foods that, oh, this is chocolate, this has 2 times the antioxidant potential. No, we worked with one company where they were, we put together a formulation of gap nutrients. So nutrients that the American public has a gap in their diet. They don't get these nutrients. We put together a formulation in like a Smartie tablet that you could have, you know, just to eat as a supplement. And we told the sources of plants that they needed to get, and they sourced these plants all around the world, and they got some bilberries from China, bilberry extract from China, which I was really suspicious because bilberries don't grow in China, but okay. So we analyzed it on the HPLC. Bilberries, you know, would have 28 different anthocyanins. This was going, so was red cabbage. They sued. They had to sue the company to get back all the money that they'd put into buying this ingredient because it wasn't there. So yeah, it's a huge problem in the industry, and very little regulation. Certainly needs it. The policy is huge. Anybody else? Please. Down in front.
Oh, I'm sorry. Really quickly, you look at the extent to which plant-associated microbes contribute to those active ingredients. So, for example, blueberries, particularly, one of the main reasons why you could get such a significant with and without their.
It is. It's enormous. It's enormously. That's part of the reason wild blueberries grow where they grow, and you can't, you know. Oh, I'm sorry. Yes. Plant soil microbes and how that might contribute to what the plant has given. It's a huge area. I don't personally, we did a work in Central Asia where we were looking at the contribution of different microbes in the soil, including endophytic fungi that went up into the plants that actually contributed quite a bit to the bioactive content of the fruit or the vegetative part of the plant that's eaten. So it's a huge area that I know very little about, but it's a contribution that is underappreciated for sure.
What about humans? We have such a big person.
That's gets in the whole personalized nutrition and the contribution of the microbiome. I would say this is such a new area. I can't answer your question, but it's there. It was really only 12 years ago that the first publications, and they came out of Canada, were showing how what was our friend Eve's Eve. Cranberry ingestion changed the composition of the microbiome, you know, and nobody is like, it was like the light going on. Well, that's how it works. I mean, seriously. And it put nutrition, it put a lot of the studies we've done up on their heads. So we're just really learning, but yes, it makes it hugely difficult to predict. We still have responders and non-responders. We do know that feeding some of these phenolic compounds will change the microbiome diversity, and diversity is seemingly always better. The more diversity of organisms, the better it's been as far as health. Sure.
Says when you make these compounds, whey protein, how in vitro do you study the release mechanism? Is it based on a pH type of a
Oh, okay. Do I need to repeat that? Okay, so in vitro digestion, how do we measure? We've done simple things like just pepsin digestion, but we've also done more complicated CACO2 cell models to look at release into the gastrointestinal tract. We have done 3-stage where we're looking at gastric and stomach and looking at the different stages of gastrointestinal to bring it through in vitro. And then we've done the animal studies as well, which are really more robust but more expensive. So just depends on what we have been able to do. But the 3-stage digestion system, and I could shoot you some, or whoever that is, I could shoot some recent publications that will show the 3-stage digestion system. And we've kind of roboticized it, made it robotic. Okay, so easier to do multiple samples with this system. Please.
Excellent presentation. One question. Okay, general question. Researchers have been doing all these clinical trials at in vitro, but there was confusion of course.
Terrible.
So when do we say we have enough evidence? Because
We have
a lot of
enough
clinical
evidence.
trials, but still people are looking for evidence. And
People are looking for evidence, and they will get excited when they hear a talk, for example, and they'll go home and they'll change their behavior for a week. So the only thing we can do is make it more palatable and make it, like I said, smaller volume. So you can get more bang for the buck in a gummy or a product that's easy to eat and not that you don't hate eating. Functional food should not be something you dread. And too often it is. That's for sure. So we're trying to kind of change the paradigm there.
Seems like a meat substitute would be a perfect way to do that if you could incorporate some of the
Like Impossible Foods, yeah, or Beyond Meat. And actually a couple of my graduate students have gone to work on that. And we haven't interacted on that. That's actually kind of a good point. We have done work with dog food, like incorporating it with the meat of the dog food, but you're probably right. That would be an easy way if you really want to, be serious about your veganism. Oh, interesting. So he's asking, are these covalent or non-covalent bonds? Mostly non-covalent, because it's the simple principle. It's medium polarity polyphenolic compounds, and they just naturally bind to bind to protein. It's the principle of tanning leather, right? Tree polyphenols on animal hides make leather. It's the same principle. It's a natural bonding, mostly non-covalent. We're just applying it to food, but we can induce covalency. We do that sometimes because it makes it even better for the allergy. Because sometimes, like you don't want the thing to break apart inside the body, and although there's caveats on that. But yeah, sometimes we do induce covalency, but mostly non-covalent.
So we're a bit over time, so
Yeah.
I'd like to present this to you. This is just a little gift from the Graduate Student Council. It's a little bag, and you can take it out to the blueberry field and stuff like that.
Thank you so much. I
There's some
appreciate
goodies
it.
and stuff in there.
Thank you so much. I appreciate it.
And there's also